just try and stop me. Okay. So in this way, with this pipeline, we can identify 98 genes. And the bottom line is that the 98 genes we identify are extremely relevant to AD biology. So taking you step by step, the genes we identify are closely related to genes that are known to be part of Alzheimer's disease based on prior GWAS studies, because in a network, they diffuse to them very, very strongly. Secondly, in a postmortem analysis of gene expression in AD brains versus normal brains, the genes we identify are statistically significantly dysregulated. Third, if you map those genes onto known transcriptomic networks from brains, then they populate pathways which are known to be involved with Alzheimer's disease, and in particular involve cytokine signaling, synapse pruning, and microglia phagocytosis. Finally, we can put those genes into models, fly models of neuropathology that overexpress tau or overexpress amyloid, and those genes modulate these phenotypes of diseased flies so that they impact neuronal neuropathology. And then finally, you can compare how well we can predict or separate cases from controls with these genes compared to just ApoE, which is the best biomarker of Alzheimer, and we can do a lot better with those genes. So all those data suggest that these genes are highly relevant to disease, so that this way of scoring the variant of unknown significance with the evolutionary reaction equation is useful. What about small samples? Well, it turns out that instead of studying 2,500 cases, 2,500 controls, we can randomly just pick 2,000 cases, 2,000 controls, 1,000 cases, 1,000 controls. We can downsample. And we downsample, we still recover a lot of the genes that we had to start with when we had 2,500 patients. And this recovery rate is much greater than if you do GWAS. So we believe we can study a very, very small sample size of 1,000, 700, 500. If the setting is right, I'm happy to push it to 200 or 100 patients. So certainly we can study about 1,400 females, 1,000 males, and many of the genes we identify were found before in the full cohort, but some of them are specific to males, some of them are specific to females. If you look just at the female genes or at the male genes, they are connected to the known GWAS genes even better than the combined genes we had before. And they predict in females the risk of Alzheimer as well as in before. But in the males, the prediction is even better. This is 0.88. Finally, if you map those genes onto transcriptional networks, some of those mappings implicate the same extracellular matrix processes and the same neurosynaptic biology processes in men and women, but some of them are very specific to men: stress response organelles, immune system, stress response organelles here, and then cell cycle biology, and one more, cell cycle biology and DNA quality control in women. So if you focus on the cell cycle biology pathways that we identify uniquely in females, those genes are related to a set of genes which we know are dysregulated specifically in female brains. The expression of those genes seems to correlate to a certain level of significance with plaque deposition in AD brains. And then again, these genes modulate fly neuropathology in models that overexpress tau or overexpress A-beta 42. So altogether, these data show that we can identify genes that are relevant to neurodegeneration that involve pathways specific to man or pathways specific to women, and that this is, we believe, a very general framework to identify genes linked to complex diseases, to identify mechanisms or enrich mechanisms, identify potential drug targets, and build risk models that are better than before. And this is robust in small cohorts, and we can do it in sex-specific fashion. So the question is, how does this really work? So basically, what I said is that we do something to coding variants based on evolution, and then we feed those coding variants for cases and controls into algorithms that then identify genes that have some sort of excess coding variants, functionally impactful coding variants in cases in control. So for full disclosure, the algorithm I just told you about is not the only one we're working on. We're actually developing an entire class of algorithms that study genes one at a time, study all genes at once, study genes in the context of their protein-protein interaction networks, study functional units. Many of those papers are already published. Some of them are still being written up. What they all have in common is that they all depend on the scoring of variants of unknown significance, coding variants, using this evolutionary action equation, and then on the fact that the distribution of this impact in normal genes and through normal variations follows a Boltzmann distribution, and that the pathogenic gene follow a non-Boltzmann distribution. Now, these items don't follow from machine learning. They don't follow from any training. They follow from first principles of physics applied to fitness landscape. So now we're going to flip to physics in fitness landscape. This work is deductive, explainable, predictive, quantitative, and testable. So first, some definitions. We define fitness as an energy potential to feed and to protect. Energy potential is the operating word. Now, a fitness landscape is a mapping. I'm sorry, I think the fonts got disrupted. A fitness landscape is a mapping F of equipotential energy contours. So this is the fitness landscape. It assigns to each genome gamma and position S a fitness value, which is the potential to do work. Now, a mutation is a move in this landscape. And when the mutation goes against a large gradient, there is pushback against it. But if the gradient is flat, 0, then there is no resistance to the move. So basically, mutational paths mostly follow evolutionary forces dictated by the evolutionary gradient. And a mutation is a move that performs the work against the gradient. So we've tied a mutation to a move, the move to an energy. So now we can do energetics in fitness landscape, provided we can measure the gradient of the fitness landscape and the size of a mutation. So this is old work from 2014, the evolutionary action equation, where we measured the gradient based on the evolutionary trace algorithm, which itself dates from quite a bit far back, and the size of a move through transition log odds. And this equation is supported by a lot of data. First of all, we've competed in the KG competitions, which, as you know, are like the CAS competitions. They're very objective. And we don't win every time at all, but we tend to be, this is the red evolutionary action, we tend to do fairly consistently well across the years. So we do very well against the best other techniques, which are all machine learning, essentially. Now, to score, we can score the impact of mutation. This is predicted on the x-axis. This is experimentally measured on the y-axis. There's a very good correlation between the equation and the experimental measurement in eukaryotes, in viruses, and in prokaryotes. And in people who have Mendelian diseases, there's also a very good correlation between the predicted impact of the mutation from the equation and the measured clinical impact on the patient. But what concerns us the most here is that if I measure all of your mutations in this room and I score them with the evolutionary action equation, they will fall along the decaying exponential that fits precisely a Boltzmann distribution from statistical physics. Whether we do it in man, in macaque, or in E. coli, the Boltzmann distribution is followed. And this is true over an entire human or population, in a single individual, in the autosomes, in the sex chromosomes, and so on. So basically, what this shows is that at steady state, our mutations follow Boltzmann distribution. The exception is mutations are known to be pathogenic, which of course they're pathogenic because they have a large impact. If they have a large impact, they have a large evolutionary action, they have a lot of energy, so they will be biased to the right side, to high scores. So this leads to the ability to distinguish benign from pathogenic mutations, but it suggests two rules of evolution that apply to all mutational ensembles, that in health, the energy of background mutations follows Boltzmann statistics, but in sickness, the energy of driving mutations does not follow Boltzmann statistics. Hence, the algorithms that we do. So we score the impact of mutations, we separate Boltzmann from non-Boltzmann, and the separation is done by those machine learning or various algorithms. Now, there's something a little bit troubling, which waving of the hand. I talk about energy of mutations, and I talk about Boltzmann distributions a little bit too freely. It is true that the distributions we find fit a decaying exponential. Are they truly Boltzmann? This is a profound question because it asks whether we truly are mutational systems that follow statistical thermodynamics in the fitness landscape. And really what I've shown you is that it is consistent with the Boltzmann distribution. I have not shown that really there's a prediction from statistical mechanics back to biology. So we need to test that. We need to make a prediction that is uniquely from statistical mechanics, let's say equipartition of energy or the virial theorem, which are profoundly based to, virial theorem is how you predict dark matter in galaxies, and equipartition of energy is how you get quantum mechanics, okay? So that's what you want to do. So without going into the details, we can force statistical mechanics onto the biological system and have to predict this quantity, which currently I call mu, for the gene selection pressure, the importance of the gene. We can do that in any species. And it turns out that all the genes which have the largest selection pressure cluster together in protein-protein interaction network. And those clusters are such that the degree of those genes with the highest importance is the largest. The eigenvector centrality of those genes, so their epistasis characteristics, are the largest. So the genes that we predict to be the most important are actually the most important in protein-protein interaction network. Those genes are also expressed in the most number of tissues. This is the correlation between the importance of the gene and the number of tissues that it's expressed in. It correlates also with the dominance of the gene, the haploinsufficiency of the gene. And in E. coli, if you knock down these genes, you get an excellent correlation with the rate of growth of E. coli. So we truly can measure the importance of the genes based on a property that is deduced directly from statistical mechanics. So there are other ways to measure the gene importance, but basically we've done all sorts of comparisons with all sorts of, all the methods out there, and we do better in 5 out of 6 different comparisons. So these data is a bona fide demonstration that we can measure selection pressure based on statistical mechanics. In other words, we are mutational ensembles in the fitness landscape, and our mutational ensembles follows the laws of physics. So first principles. So we want rules of biology. Those are rules of biology. So the last step is to say, if we can measure selection pressure, maybe we can measure selection pressure in genes in patients with Alzheimer and selection pressure in people who do not have Alzheimer, and see if there's a difference. And we find some genes that have a systematically significant difference in selection pressure between Alzheimer and non-Alzheimer. Well, I don't want to sound like a broken record, but these genes are very closely related to GWAS genes. They implicate pathways which are important to Alzheimer, and they discover new pathways that are important. The single most important thing is that we can use these genes to separate cases from controls. So what we did is that we took 2/3 of our entire cohort. We used selection pressure to identify the genes that are different in cases and controls. In this 2/3, we trained in this 2/3 a machine learning algorithm to separate cases from controls, and then we applied that to the remaining 1/3. And the area under the curve we get is 0.83. This is state-of-the-art, based on coding variant, based on the germline, at birth. These individuals have a huge odds ratio of developing Alzheimer. These individuals are essentially protected against Alzheimer. So this is not yet perfect separation of the distributions, but it is really state of the art. So these data show that we have algorithms that can identify really genes that are directly involved in Alzheimer's. The key thing is that we don't need very many patients to do that. This is very general. We've also done that in Parkinson's. We're doing that in diabetes. We're doing that in heart disease. We're doing that in cancer, to look at cancer germline in the breast. But there's just not one way to do it. There's a whole class of algorithms that can use those ideas. And the key, though, is that the key information is to we gain power by using more evolutionary information. That's where we get our information. And we use that evolutionary information by annotating as well as we can the impact of coding variant and tying that to the physics so we can know exactly what to look for, which is the Boltzmann versus non-Boltzmann characteristics. And most of all, basically, genetic information obeys the laws of statistical mechanics. So in this way, we can weigh gene selection pressure. We can identify genes, mechanisms, risks, and drug targets. So this is a way to essentially unify physics, math, with biology and evolution. So this is the work, of course, of everybody in my lab. Thank you very much.
Thank you so much, and I apologize for the technical difficulties.
Oh, not at all.
And then thank you to our volunteer for swiftly fixing those. So, questions?
Thanks, great talk. I was wondering, you worked on exome data, correct? And your evolutionary trace method works on exome sequences. Can it be extended to non-coding variants?
Gosh, we're trying so hard right now to do that. The main difficulty with extending the evolutionary trace to non-coding variants is to get good alignments. Now, I do believe that people who work on ENCODE say they have really good alignments of regulatory elements. They even have alignments across species. So if that's true, that would be really, really wonderful. The other possibility is to use those ideas from statistical mechanics that I used and that I mentioned and try to apply them directly to non-coding variants. And that's the second path we're trying to take right now. I can already tell you that looking at splice variants, we see hints that this will be useful. In terms of risk prediction, your question is terribly, terribly important because to identify the genes, the coding variants are great, but to really predict risk at an individual level for diseases that are highly regulatory based, then we really need some more information on coding variants. So it's a limitation we're working on. Thank you very much.
Thank you.
So one more question because of time.
I'll be brief. You said something very intriguing. You said there's a different selection pressure.
Yes.
On variants in disease people as opposed to non-disease people. I don't understand that because that just doesn't make sense. This is Alzheimer's. Everybody has already reproduced by the time they have the disease. So where's the selection pressure coming from?
It's a semantic issue. You are correct. I'm not disputing what you're saying. What is happening is that I am selecting a set of individuals which have Alzheimer and selecting a set of individuals which do not have Alzheimer. So I'm essentially taking them from different parts of the fitness landscape. And in order to inhabit their respective parts of the fitness landscape, they have a differential set of mutational energies in specific genes. So in that sense, if you just look at those different populations, it just apparently translates into those genes being under different selection pressure. And that's just because as an artifact of me having chosen to label them correctly based on their different clinical phenotypes. So if I took people who are over 7 foot tall and people who are under 4 foot tall, in the same way, I could identify genes that are under selection pressure to be tall or to be not so tall. Yeah. So it's a slight semantic issue, but there's nothing contradictory there.